Okay, we'll get started here. It's a little bit after 109 now, so a little bit after 108. Usually about when the last people trickle in. Not to mention any names. Some of you guys who are the last ones every time. Aiden, hi. How's everybody doing? All right. Well, just sending out the last couple things here. I know you guys probably know this is the last week. I put in grades. Took me a while to decide how to do it. You guys have been doing a pretty good job, good attendance, stuff like that. If you want to know how it works, we were kind of instructed by administration teachers and stuff that they discussed, I guess, at a pay grade above mine, how we were supposed to do grading. So it's kind of like a full completion, partial completion, or didn't do it at all. So you kind of see the way I put in your scores. If it was like a big one with a bunch of IXLs, I made it worth like 5 points each. So 9 times 5, 45 for that. For a different, shorter assignment with just a couple questions, it would be worth like 10 points. So again, you either basically got 100% if you did it all. You're still able to complete stuff. So a couple notes that I took from people who emailed me. Yeah, you can still finish the IXLs like the R3 through 5, like the ones on the adaptations and natural selections. Those ones I kind of talked about. I did the first two and like looked at those, but I hadn't tried the other ones, and I didn't know if you guys had to do like hard math because it had to do with percentages. I didn't know if you had to do a Hardy-Weinberg equation, so I didn't assign those right away. But then I looked at them and I realized they were super easy. Well, not super easy, but they were pretty easy. So I went ahead and assigned them. But a lot of people didn't get those done, and you asked me questions about it. Yes, you can still finish up any of the IXLs. For the people who asked me the questions about those videos for that one Google form, that attendance deal. What was it? The primate and human evolution videos. Like some of you guys said the links didn't work. Well, do it on a different computer. For some reason, Chromebook is blocking them. You can type in the title. Last option, and I actually said this in the other meeting because I was actually rewatching one of my meetings, is just watch it on your phone. Like your phone, when it's not connected to internet, just the title of those videos. So a couple people said that they hadn't submitted those because they couldn't get the links to work. Well, if you just watch it on your phone and then fill out the summary that matches the video and just submit the form, that's all you have to do. Okay. The last thing, if you're wondering what I did for, let's see, what do we got here? Oh, of course. Let's see. The geologic time research slides, I am just going through and trying to find everybody's. Some people shared them with me, some people emailed them to me, things like that. So if you haven't got a grade for that, it's because I just haven't got to it yet. So if it was like in my shared folder and I marked you off, fine. The rest of you, I don't know. If I can't find it, I'll email you and ask you to share with me that, because it takes forever to look up everybody who shared me something and then look through their thing. So open up the first one, spend a couple minutes looking through it, open up another one, spend a couple minutes looking through it, open up another one. It just takes a while. So that's why you haven't got a grade in there. I've only looked at a couple, and just searching my inbox takes forever. Okay, that's enough about grading notes. Anything about that? Does everybody kind of understand what they had to finish up? So I tried to, yeah,
Mr. Jones.
yeah, fire.
So it says that you're missing my primate and evolution thing in Power School, but I completed it and I turned it in.
Okay, are you the ones you emailed me, right? Because I'm not very smart sometimes, and I like when you go to the form, it doesn't always collect email addresses, and I had you send me that email, right? Because I didn't
Yes.
collect your email. So I forgot about that. So I just have to go mark you off now that I'm going to write it down right now. And I can't remember who the other, I think it was maybe Morgan and Brooklyn were the other ones or something like that.
Lily was one of them.
Ah, yes. Perfect. Ah, yes. If anybody else, it was the first, and then I resent it back out, and then it collected email addresses for everybody who sent it after that. So I have a pretty good idea. I can kind of process of elimination. Like there's 3 or 4 people who I don't know if I'll hear from, but then the rest of you, I can kind of, based on your answers, I can figure them out sometimes. Like if you Snapchatted the answers to your friend and they're the exact same, that kind of stuff. I mean, I can figure. Okay. Let's go. What do we want to talk about here after this? Gosh. All right. Next thing is brainstorm stuff, and everybody should have got the Master Kingdoms thing. So Usually, like, we spend, I don't know, 2 weeks on kingdoms and classification. We talk about the 6 kingdoms of life. We break it down into prokaryotes and take care of like archaea and eubacteria or bacteria. That takes like a day. Then we go on to protists and fungi. That takes a day or two. And then we go on to plants for 3 or 4 days, sometimes as long as a week, and animals for 3 4 days, sometimes as long as a week. And then we dissect pigs, and then we do pig dissections and systems and stuff like that. So um, to do this all together at once here, we usually do kind of like this little brainstorm. So if you guys haven't seen it yet, I'm going to do a little bit of a screen share, and I'm going to ask you guys some questions, and I want you guys to be prepared to fill in this master, let's see, Kingdoms Overview Brainstorm. Here we go. I got to get to my screen share. Sorry, I'm clicking around. There's people bumping in and out of the meeting. I'm always looking at the chat, so it takes me a second. Let me get to screen share, and I'll pull up the brainstorm, and you guys can help me. Just, I have to assume that some of you have looked through the notes a little. Maybe you saw the form I just shared with you, and you can keep filling that out as we talk today. So open this up. And we're just going to hope that some of you guys have looked at this so we can actually get through it. All right, you guys seeing my screen? Let me bump this up a little bit bigger. Hey, look at there. There's some people in here and everything. Wonderful. All right, so when it comes to describing some of the defining characteristics of each of the 6 kingdoms, that will help you answer some of the form questions, and it'll help you fill out this thing as well. So let's talk a little bit about, it says cell types and number at the top, and then it says nutrition and metabolism, then reproduction, and then some defining characteristics. So these are your 6 kingdoms, okay? 2 of these kingdoms are prokaryotic kingdoms. The rest of these are eukaryotic kingdoms, and they have some defining characteristics. So archaea vary greatly. There are ancient bacteria. They're extremophiles. They live in sulfur vents. They live in acid caves. They live inside of the guts of termites. They're just super extreme versions of prokaryotes. The rest of the prokaryotes are called just eubacteria, or sometimes just called bacteria. So the reason we don't always say the U in front is because that means true bacteria. So archaea were separated from the bacteria into 2 separate, not only kingdoms, but domains, okay? Domains of life. So those are our classification systems. So I'm looking here at my forum as I'm talking about some of this stuff. So as you guys are looking at this, cell type, what could we put for archaea and eubacteria? Lily, are they prokaryotes or eukaryotes, both the archaea and eubacteria, based on what I just said?
Eukaryotes?
No, they're prokaryotes. Okay, so both the archaea and eubacteria are both prokaryotic. Okay, meaning they don't have a nucleus. Okay, that's the main thing that prokaryotic means before the kernel. Prokaryote means before the kernel. So if someone wants to type in there that the cell type of both archaea and eubacteria is prokaryote, that'd be fine. Now, I also have that number. Okay, do you think archaea and eubacteria are mostly single-celled or multi-celled? Let's see, Brady, real brain buster here.
Single.
Yeah, prokaryotes are almost predominantly all single-celled things. Now, there are some bacteria that live in like colonies where they kind of divide up labor. So even though bacteria are super simple single-celled organisms, they sometimes make like masses that almost act like a colony. So we say that some bacteria and some archaea are nearly colonial, meaning that they have a division of labor amongst a group of single-celled organisms that live really closely together. It's kind of like mutualism. So I guess for a cell number for both archaea and eubacteria, they're basically all single-celled. Okay, so that must mean that all the other ones are eukaryotic. So protists, the fungi, the plants, the animals, those are all examples of eukaryotic kingdoms, meaning protists, fungi, plants, and animals have cell nuclei. Okay, along with that, they have all kinds of other organelles. Okay, plants have a chloroplast. All of them have mitochondria and maybe vacuoles and endoplasmic reticulums and all kinds of other structures as well. Okay, so the other 4 kingdoms, okay, they vary super greatly. So the protista is kind of like the junk closet eukaryotic kingdom. It's where we put all the weird things. It's where we put like algae. They're not quite plants. but they're not anything else. Okay, that's where we put protozoans. They're not quite animals, but they don't fit anywhere else. And that's where we put slime molds. So plasmodial slime molds, like they're not quite a fungi. We don't have anywhere else to put them. So you can see that our classification system is flawed. Okay, so the first question in the forum is about how has our classification system evolved over time. You guys are welcome to look through the notes and read all the details about the changing of, the evolving classification system, and just kind of summarize in your own words from, like horrible ideas of what life was to now a modern, I guess, phylogenetic DNA RNA based system where we can kind of classify things at the molecular level. Okay, so what are the two main types of reproduction that we see in the world? So I'm looking at my forum and I'm trying to keep my pace here. So let's hear from Abby Pieper. What are like the two main modes of reproduction? Okay, just as kind of an overview. Like what are the options basically as a whole to reproduce and create more of your own kind?
I don't know.
So with or without sex, we just have to put something in front of it. So we've got sexual reproduction, and what's the opposite of sexual reproduction? Without sexual. So we put the letter A in front. So asexual. Okay. So if we were to, if you guys, some of you guys can keep filling some stuff in here as we talk about this brainstorm. So all these other four are all eukaryotic kingdoms. That'll help you with the form if you guys are following along with my brainstorm here. We're going to talk about nutrition. Yeah, there we go. Okay, so we're going to talk about reproduction. Now, most organisms don't actually have sex. Okay, there isn't even sexes. Okay, so there's not boy and girl bacteria. There's some weird stuff that happens with protists. Fungi are crazy. Instead of just being like two genders, there's like 14 different sex types in certain types of fungi. So sex gets really complicated, but in general, we can either say an organism reproduces sexually or asexually. So prokaryotes, the archaea and eubacteria, their form of reproduction is basically binary fission. Okay, what is binary fission? A bacteria basically copies its DNA and then splits in half. Like that's it. Okay, so we call that binary fission. It's a form of asexual reproduction. But what are they capable of doing is swapping genes. So a lot of bacteria can like trade genetic material with other bacteria, sometimes by eating them, sometimes by absorbing it from their environment, and sometimes bacteria even hook up these little sex pili where they can swap genes. I like that. They can gene swap. Yeah, actually MRSA bacteria, the type that causes staph resistant and antibiotic resistant strains of bacteria, they actually are able to trade basically like cheat codes to how to like beat antibiotics. So pretty crazy. So we see some, what do we call it, lateral gene transfer or horizontal gene transfer between different species of bacteria, where they literally swap genetic information that makes them more resistant to antibiotics or death in general. Okay. Some defining characteristics. So I'm kind of bouncing around a little bit here on this as you guys, I was hoping you guys would, I had a master plan. I thought you guys would kind of fill this in as I talked about it, and then we'd kind of go to the form questions. So I'm answering the form questions while talking about this as well. So yeah, when it comes to archaea and eubacteria, they basically do asexual binary fission, but they are capable of gene swapping. All the rest, except for plants and animals, usually do a variation of both. So some protists, just like most of your protozoans, like animal-like protists, like euglena, amoebas, paramecium, they just go through like mitosis and split in half. So they basically just go through a very primitive form of asexual reproduction. Yeah. Thank you, Jordan. Okay, as people are filling some stuff in here. Fungi, again, some fungi are asexual. Most of the yeast that we use to make like breads and cheeses and wines and other yeast and stuff like that, most of that is asexual. Most of those just basically do the same thing. They just copy their DNA and divide and split their nuclei. But there are a bunch of fungi that have spores, sex spores. So your most common type of so right now is morel season. So a lot of people think that a morel is a mushroom, but if I haven't said this yet this year, I'll say it now. Morels are not mushrooms, they're ascomycetes. Okay, so the difference between a mushroom like a portabella, that's what we call a club fungi. Okay, it has like a cap with gills underneath, and a morel and a morel has kind of like, I don't know, morels look really weird. Okay, those are what we call cup fungi. So their spores are produced in little cup-shaped structures. So you're not technically mushroom hunting. You're fungus hunting, and a morel is a fungi, but not technically a mushroom because it's not a basidiomycete. Instead, it's an ascomycete. So that was fun. I'm hoping you guys enjoyed that. Okay, plants and animals do a variety. So most animals use like sexual reproduction, and a lot of plants use a form of sexual reproduction. But plants are weird. Plants do alteration of generation. So they have a sporophyte and a gametophyte generation. So the plant is able to reproduce both sexually and asexually. And then there's every variety in between. So you can cut up a potato.
Shorts to be at home.
You can cut up a potato and throw it in the ground and grow potato plants. You can make clones of that potato. You can actually cut off.
Can you really?
Yes. So you can use roots and tubers and different types of. Yeah, sure. Take a potato, cut it up, bury it. You'll grow to the potato plant. You didn't know that? Okay. There's other stuff. I got a little sidetracked there. And somebody's pulling in my driveway. Very distracting. All right. What else should I say about plant reproduction? It varies as the plants get more and more advanced. So like mosses don't have seeds. They're non-vascular seedless plants. So in order for a moss to reproduce, it has to get wet. So the Antheridium, the Archegonium, you have to have a wet pad of moss, and then the sperm of the moss actually swims across the water film and fertilizes the female. And that's how plants can do a combination of reproduction. Animals, most of them use sexual reproduction, but think of a simplest animals possible, porifera, also known as sponges. Sponges, you can basically chop a sponge up into 5 sponges and you get 5 sponges. So sponges we call that fragmenting. So you can take a part of a sponge, cut it off, and grow a new sponge. So again, that's kind of like a potato. Okay, defining characteristics of the different ones are kind of vary greatly. Eubacteria, they all have a special protein or glycoprotein in their cell walls. All eubacteria have to have peptidoglycan. That's a rule. Okay, so in order to be classified as a eubacteria or what we call just a bacteria, they must have a specific chemical in their cell wall. Okay, and that chemical is peptidoglycan. Now, archaea have way different types. So some archaea live in like the Dead Sea, where it's just insanely salty. Other archaea live in like sulfur vents. Okay, and some live like way deep in the ocean, and they do all kinds of crazy stuff. So they're just like, again, the junk closet of the prokaryotes. So archaea are kind of the junk closet of bacteria, and protista are kind of like the junk drawer or junk closet of your eukaryotes. So there's just a whole bunch of different stuff that we can cover here. Okay, let's go through and look at some of the stuff from the form. What do I mean by, nutrition and metabolism? Before I go on, let's talk a little bit about this. So what are the 2 main ways that you can produce energy from your environment? What are the 2 modes of nutrition and metabolism? Morgan, I haven't heard from you. I just kind of scroll across my screen and then pick whoever's there. Morgan, what are kind of the 2 ways you can kind of get energy?
Like from like foods.
From what?
The sun, like.
Yeah. So we call those autotrophs, right? So autotrophs are things that can do photosynthesis or things that can do chemosynthesis. So an algae is an autotroph, right? But a plant is also an autotroph. Okay. But there's also some bacteria called cyanobacteria that can also do a form of pretty much photosynthesis. So yeah, forms of nutrition vary very greatly across. So where it says nutrition and metabolism, under these, it kind of depends on the kingdom. So you'll notice that our classification system is wild and somewhat confusing, okay? Because there's only some consistent rules, okay? So these ones are single-celled prokaryotes. These ones are eukaryotes. Some eukaryotes are single-celled. some are single-celled and some are multi-celled. So some protists are single-celled, like a paramecium is a single-celled protista. Okay, but a kelp, which is a type of algae, a kelp is a giant aquatic photosynthesizing 40-meter tall thing that lives underwater. Okay, so a huge variety amongst these characteristics, but there's some defining ones. Okay, so Morgan told us that you could be an autotroph and do something like photosynthesis or chemosynthesis, like things that live in ocean vents, or you could be a heterotroph. And there's all kinds of different types of heterotrophs, decomposers, scavengers, but those aren't very common types. The ones that you guys should probably know are herbivore, carnivore, and omnivore, different types of heterotrophs. Okay, so again, you've got just massive amounts of diversity, not only between each of these kingdoms, but within. Like within Protista, it's just a crap show. Okay. Within Archaea, there's just a crap show. Okay. Within fungi, there's a whole bunch of different stuff, but there's some universal things that make a fungi. A fungi has to have chitin in its cell wall. It has to do extracellular absorption of nutrients. So whether it's a mold or a fungus or a morel, okay, or a portabella, they all basically grow a bunch of filaments on food, and then they excrete some enzymes on that food, and then they absorb some energy from that food. So if I'm looking at the mold on an orange, or I'm looking at a mushroom growing, I don't know, in my yard, or a morel in the local, I don't know, deciduous forest we have around here, they all do the same thing. So again, the point of this is to understand the diversity, not only within, but also between these kingdoms. Okay, so again, I have got to manage my time a little bit. So let me get to this. Come on, get out of here. No. All right, that was lovely. Worked really good. All right, so you can use this to answer the forum questions, but let's kind of go through them. Okay, so here's the classification, history of classification. Oh no, not people trying to get into the meeting. All right. So first question is about just a summary of the history. So like Aristotle gave us some common names and generic names of things that we still use the same, because they used like Greek and Latin back then. So some of the names stuck. So he actually named a bunch of stuff that we still like use the same Greek or Latin name. It's just kind of modified from language to language. So Aristotle was big into classifying. He was just really bad at it. He just wasn't very accurate in his form. John Ray's the species guy. We already talked about that. Van Leeuwenhoek is the guy who invented a microscope so he could see little things. Here's Linnaeus, the father of taxonomy. Guy like Haeckel basically discovered Protista. Again, you guys can summarize this on your own. I'm not going to go through each and every scientist, but it goes all the way to Whitaker and Woese, okay, the two that you had to answer in the previous one. So going from 2 kingdoms to 3 kingdoms to 4 kingdoms to 6 is what we have now. Okay, here's your 3 domains. Okay, bacteria or eubacteria, the archaea and the eukarya. Here's how that fits on what we call the phylogenetic tree of life. Okay, we can see our 2 prokaryotic kingdoms and then our different groups of eukaryotes over there. Here's the taxons that we talked about. So you guys have already listed these a couple times. Dumpkeep King Philip came over from Great Spain, helps us kind of break things down. Here's a picture of my adorable son when he was younger, and just going through Domain Eukarya all the way up to him being a Homo sapien. So again, you can break down the classification system. I could spend however long on that. We still use Linnaeus's binomial nomenclature today in modern taxonomy, but we use better molecular genetics in order to classify things. Cladograms and trees are ways that we put things in groups, and we separate them by traits that they share from traits that separates them out from one another. So synapomorphies are things that everybody in the group shares, and plesiomorphies are things that help divide or help us split off different groups. So here you can see a cladogram of vertebrates. So amphibians do not have an amniotic egg, but they share a vertebra. They share a backbone. Okay, These ones, turtles, don't have an opening in the skull, but everything past here. Okay, these things all here, they don't have hair, but these things up here do. Monotremes is a type of mammal. Marsupials are a type of mammal, and placental mammals are a type of mammal. So we can see that there's different things that help us. These are all vertebrates, but what separates amphibians from reptiles and turtles, crocodiles, birds, and dinosaurs, all kinds of different characteristics that we use. Okay, so these are modern cladograms and phylogenetic trees. Okay, moving on. All right, 6 kingdoms of life. There's 2 prokaryotic kingdoms. There's a whole bunch of ways that you can see that it's kind of a crap show. So there's major ways of just separating the kingdoms, and then it breaks down into even more specific ways of breaking down the kingdoms. So prokaryotes versus eukaryotes, that's where we start. And then after that, we start talking about things like, how do they get their energy? So what is their nutrition and metabolism? How do they reproduce? What is their form of sex? Okay, do they do it asexual, or is it sexual, or some variation of that? Okay, how many cells do they have? Are they unicellular, multicellular? What type of cellular arrangement is there? Okay, so here is Volvox, which is a type of alga that makes a colony. So this ball here could break apart and all these cells could live individually, but when they work together, they make life easier for themselves. Okay, this right here is just dividing yeast cell. It's a type of fungi that's just single-celled, so these aren't working together. It's just this one becoming 2. And then, of course, there's an African elephant, and that's a giant multicellular animal with organ systems and all kinds of complex things. Okay, these are all living things, but they're just crazy different from one another. So we have to have a way of classifying. Humans have to classify stuff. It's the only way we can make sense of the world. So autotrophs and heterotrophs and reproduction is something we've already talked about. I'm trying to go through the questions. Different types of heterotrophs right there. Okay, again, advancing our classification system to our current one. There's a very nice chart for helping fill out the brainstorm. There's a nice picture. We talked about life's origins already. Here's another very simple diagram of the tree of life. All right, let's get to some more questions. Again, I talked about binary fission for bacteria and lateral gene transfer, which we talked about. So 3 different ways that can happen. I can't remember if that was a question or not. Let me look. All right, so I've covered most of that stuff. I'm just kind of going through what I've covered and what I haven't. So again, look at the prokaryote stuff. How do we classify bacteria is the next question that comes up. So like bacteria have not been known for all that long. Like germ theory is not that old. Like we used to think it was like, I don't know, demons and spooks and bad omens and stuff like that made people sick and die. Nowadays, we know that it's caused by microorganisms, pathogens, antigen agents, things that make us and harm us in some way. So really, the bacterial classification was just basically developed as a need to help us start treating people who were dying once we figured out people were dying of like tiny things we couldn't see. So there's all kinds of different ways. So one of the big things is like gram staining. Okay, so a serotype and gram staining are kind of similar. It kind of tells you what it is. So a coronavirus is an enveloped virus. It has a corona or crown, and it has a bunch of like little proteins that hook up to our cells and then let the virus inside. Okay, that's a virus, not a bacteria. Okay, gram staining, so gram-positive bacteria versus gram-negative bacteria. It's an important way that we separate groups of bacteria from each other. That will say, hey, should we use penicillin, amoxicillin? What should we use to treat this person who has some sort of infection? So anaerobic versus aerobic. Can they live in oxygen or can they not? So tetanus, okay, if you get a tetanus shot, basically they form like an endospore on like a piece of metal. So let's say that there's a bacteria that lives on a metal sheet. So RJ's out in the barn right now and there's some tetanus endospores hanging out on a piece of metal, okay, like a nail. And let's say he jabs that into his body. Okay, that endospore could wake up and basically start doing a form of reproduction and respiration inside of his body, releasing an exotoxin, okay, that would cause severe muscle cramps, spasms. As a matter of fact, with really, really bad tetanus, the bacteria will release a toxin that would make you spasm so hard you would snap your own back. So tetanus, bad. Don't want it. We can classify bacteria by their shapes and arrangements. So these ones people have probably heard of. Hey, do you got strep throat? Hey, do you have staph infection in the cut in your leg? Okay, so cocci, staphylo, diplo, all kinds of different ways that we describe bacteria based on their shape and arrangement. So again, the bacteria classification is crazy, and it's based on what's most practical. Are we using this in medicine? Are we using this in classifying, I don't know, some crazy organisms in a cave that we just discovered? You're going to use a different system. They get around a lot of different ways, but they're super important. So we may not appreciate our bacterial and prokaryotic friends, the archaea, but they live inside us, among us, and all around us, and they help basically with the world. So why, how, okay, some stuff down here. Okay, they're super important to the food chain, nutrient cycle, decomposition, the pharmaceutical industry. Anybody who has diabetes, we already talked about this earlier. You can actually insert the human gene for making insulin into a bacteria, and they now make us insulin treatment for diabetics. Okay, we have bacteria in our guts that help with digestion. If a cow, any ruminant, okay, anything that like does cud chewing, anything that has to swallow something super high in cellulose, break it down, puke it back up, chew it, swallow it again, they have bacteria in their guts, mostly methanogens that break down cellulose and release methane. And that's why cows burp and fart so much, because the bacteria that live inside of them help them break down stuff. All right, sorry, this is painful. Hopefully not as bad. Hopefully not too bad. I'm trying to answer the questions for you guys. The protists are broken down into 3 major groups. It's listed right here. Animal-like protists, plant-like protists, and fungi-like protists, which we call mycotas or slime molds. There's a lot of names for them. So protozoa, from the word zoo, means animal-like protist. Algae, everybody knows algae. Okay, it's the stuff that grows like seaweed and kelp and I don't know, sea lettuce and other types of algae. Other things like diatoms that are in like toothpaste and cosmetics and things like that. All kinds of algae that do photosynthesis. Okay, and then the fungus ones are really crazy cool. As a matter of fact, they're even like almost smart. What do I mean by that? Well, there's a video on that. There's just a bunch of pictures of different ones. Here's some of those cool slime mold videos I was talking about. It talks about the different types of Myxomycota and Ascomycota and how they have really weird life cycles and all kinds of crazy stuff. We don't have time for that. Ain't nobody got time for that. Euglena, that's a crazy protist. Euglena actually can spontaneously develop chloroplast. So if it prefers eating, so if there's lots of food around, so if there's lots of other protists or bacteria for the euglena to eat, it will eat them. But if the euglena eats up all the resources and it doesn't have anything to eat, it will literally turn on a gene that it has hidden in its genome and start building chloroplast. And then it will start acting like a plant and will start doing photosynthesis. Crazy. Okay. Oh, some diseases of protists. Dysentery, if you've ever played Oregon Trail, or if you've ever drank bad water, when you watch like Bear Grylls on some show like that, or Naked and Afraid, if anybody's into that weird show. Okay. Yeah. Don't drink the water. It's full of little microbes that can make you have explosive diarrhea until you get very sick. So there's different types of there's amoebas that cause dysentery. There's amoebas that actually eat our brain. There's amoebas in water that cause giardia or beaver fever. There's African sleeping sickness, which is transferred by flies. Okay, so the tsetse fly gives people basically a very bad disease that's often fatal. Malaria has probably killed more people than anybody in the world. So like, that's kind of a bad protist. And then toxoplasmosis. So pregnant women should not have kitty litter out. That's bad. Okay, so that's just some. Holy crap. We got to keep going, right? Okay, what about fungi? What's the major thing that fungi have to have in their cell wall? It's right here. It's chitin. Okay, the way fungi work is they got a bunch of hyphae, which makes mats and mycelium, and then they basically grow reproductive structures. So little things underneath the ground grow out of some spores, and then it spreads out, and then it grows a mushroom above the ground. Okay. Or how about ringworm? So if you've ever had ringworm, you got sheep or cats or something like that, and you rub up against them nice and good. Okay. Some of those spores are going to stick to you. They'll crawl into your skin. They'll start creating a mat of hyphae that eventually becomes a mycelium. And when the ringworm gets to a certain size, it shoots up little spores that stick through your skin in the shape of a ring. The size of the ring determines how big the infection is. If you have a giant ringworm, that means you have a big colony of fungi living underneath your skin. Pretty crazy. We basically classify fungi by their sexual behavior and sexual structures. So the major phyla are listed right here. You guys can look through them. That's one of the questions. There's flagellated, yeah, I don't even want to say that word right now. Zygomycota, chytrids, okay, Ascomycota. Remember, these are the ones that are morels. Again, they're different than club fungi, which are mushroom smuts and rust. That's your Basidiomycota. Those are the club fungi. And then there's some other one, Glomerulomycota, and then a weird junk closet of fungi imperfecta, which is just where we put the rest of the phyla of fungi that don't fit in with the other ones. Moving on. So again, there's kind of your different ones. Here's where it separates morels from club fungi. So you got your Basidiomycetes and your Ascomycetes, and then some of your other groups right here, which are way more confusing. Okay, but I always like to use that example. You can look through this. I already answered the questions for you. Plants kingdoms. Hey, what are the 4 advancements in plants? I'm going to slow down and I'm going to ask somebody a question. Is Levi even paying attention, or is he like is he out working for his dad and he's like, I don't know, fixing the planter right now? Levi, what are the major advances in plants?
Non-vascular to vascular, seedless to seeds.
All right. So non-vascular to vascular, seedless to seeds, and then ones that have a naked seed to those that produce fruits and flowers. Okay. So I know that was just kind of me like pausing and giving myself a chance to recollect my thoughts. So here's a nice look at how the plant kingdom works. Your simplest non-vascular plant starts off with like your mosses and hornworts and liverworts and very, very primitive, barely not algae. Okay, so from algae, we've moved on to land plants. So in order to go from the water where algae live to on land where plants live, there had to be some advances. Okay, so one of the first things was the development of vascular tissue. So mosses live on the north sides of buildings where it's wet and shady. Okay, they have to get wet to reproduce. So they have to stay close to water. They can live on land, but they can't, if they are out in the beating dry sun, they'll just dry up. They can't survive. Okay. Then you get to your club mosses and ferns. Okay. And club mosses and ferns have vascular tissue. What are the 2 types of vascular tissue ag kids out there? Ferns developed what 2 types of vascular tissue? It's a side note. It's not even one of the questions, but I hope you guys know this. One starts with X and the other one starts with PH.
Xylem and phloem.
Thank you. Xylem and phloem. So from your mosses and hornworts and liverworts, we start developing primitive vascular tissue. So ferns have xylem and phloem. Xylem is transferring water, and phloem passes around nutrients and photosynthetic products, as well as saps and other things. And we start to develop more advanced. Ferns are also seedless, just like mosses are. They got to develop like a sori. They have to, like a little cup of spores that have to get transferred. So you have what's called the prothallus that germinates and becomes the fern and stuff like that. So we're talking about gametophyte versus sporophyte dominant plant generations, but I don't have time for that. So then after that, just looking at these, we go from ones that aren't vascular to ones that are vascular, and then to ones that are seeds. So what are the different groups of, let me see, I'm going to ask somebody else a question. I feel bad for Levi. Let's see. What are the 2 major groupings of the plants that have seeds? I'll give you guys a second to kind of look. And I feel like Brady knows this. Brady, what are our 2 types of seed plants? Plants that have seeds. Ones that have dry or naked seeds, and then ones that have a seed that's coated in a seed coat or fruit. Those ones also make flowers.
Conifers.
Yeah, so you got gymnosperms and you got angiosperms. So a lot of people use the term conifers to talk about the naked seed, but conifers isn't always necessarily, they're cone-bearing trees, but in general we call them gymnosperms because we got some of our scale trees and other primitive gymnosperms. Some are extinct, a few are left around, but the majority of them are conifers. Yeah, so they're cone-bearing trees. So the cone that you find on the ground that you throw at people, that is actually the female cone. And then high up on a coniferous tree, you're going to find the male cones, which are just little tiny things covered in that yellow dust. That's pollen. What is pollen? It's tree sperm. It's the male reproductive cells. So if you've got an allergy to pollen, that's why, because they're little tiny spores that blow through the wind. And if it bothers your immune system, your body overreacts and voila. Okay, so again, we've got gymnosperms, and then the flowering plants are called angiosperms, and they produce flowers and fruits. They evolved along with, I don't know who did the periods research on this. Some of you guys might have to resend me their periods research, but as we get flowering plants, we get pollination and insect evolution and all kinds of crazy new fruits and flowers. Okay, so what are the two types of angiosperms? Kennedy, are you able to answer? I know that the internet is not great at the farm, and I've seen you popped in and out. You can chat me if you can't use audio. Oh my gosh, look at the time. I can't find out. I'm trying to look if she's chatting me. Oh my gosh. All right, no, Kennedy's really smart, and she answered that correctly. The two types of flowering plants are monocots and dicots. So monocotyledons and dicotyledons. Earlier this year, I was asked to do that little thing where we grew the corn plants in class, and it was kind of a good use of our time, maybe. Okay, kind of threw me off for a couple months there doing that thing, but I thought it was useful for us to do that little activity. But we grew some corn plants. So corns are a monocot. What makes a monocot different than a dicot? Well, vascular bundles and rings are in it's like a oak tree. If you cut it down, it has rings. Okay, because each year it grows a new vascular bundle outwards. That's what gives it a cork cambium and a corker and bark around the outside. And that's how, when you cut down a tree, you know how old it is. That's what a dicot is. While something else that's a monocot, like grass or corn, doesn't make those vascular rings. All right. There we go. Comparisons between monocots and dicots. Next question from your forms. I'm just glancing over the form as I go through this, and I know you guys are probably sick of this, and it's almost 2:00. Okay. Animal kingdom stuff. If we have one more meeting in this class, we usually finish up the year with talking about animals and the different phyla of animals. There's 9 major phyla of animals, and then we break down the major things. And then we spend quite a bit of time talking about vertebrates. And then we take some fetal pigs, which are very close to us anatomically, and we learn about fetal pig anatomy and the major organ systems of the body. And then in class, we would dissect fetal pigs. So what are animals? Well, animals are eukaryotic multicellular heterotrophs. Okay, so again, if you were to go back to the brainstorm, what are the major characteristics? They're multicellular, they're eukaryotic, and they have to like move around and eat or have some way of like absorbing or eating some food somehow. Okay, most move around, some are sessile, others have different things, but we classify them based on a bunch of different things, like how advanced their tissues are, how many body cavities they have, how many embryonic germ cells that they form when they develop, okay, from an embryo. Is there an amnion? Do they have a notochord or a vertebral column? Those are all ways that we classify animals. So it's a very diverse group, starting with the lowest animals or the most primitive ones. Choanoflagellates probably evolved into our most primitive porifera, which are sponges. So right here is a classic cladogram or phylogenetic tree of your 9 major phyla of animals. sponges, jellyfish and anemones, flatworms. Most of these are really small and cause weird diseases, but the other ones, like the flatworms that live in the ocean that are super colorful, are awesome. These are called platyhelminths. Nematodes are roundworms. So again, when you learn about, I don't know, ag or watch it, most nematodes are actually good for the soil, and they're good for plants, but there are a couple, parasitic pathogenic nematodes that you want to keep out of your fields. Mollusks come in way different forms. So mollusks go all the way from like bivalves, like this guy right here, to squids and octopus. Annelids are earthworms, segmented worms. This includes leeches as well. Arthropods is gigantic in terms of a group of animals. It includes oh my gosh, so many things in there. Arthropoda means jointed foot. So that's everything from insects to crabs to lobsters to all kinds of things. It's by far the biggest animal group. It's like beetles. Beetles alone is like half the classified things on Earth. Okay. Echinoderms would be the next things. And this would be like urchins and starfish and other things like that. And then finally the chordata. Oh my gosh, we're finally to us. Okay. And when I'm talking about us, we start out with lancelets and tunicates, and then we work our way up through the different types of vertebrates, from, I guess, amphibians to reptiles to blah, 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 blah. And then finally birds, mammals. Okay. I didn't need a blah, blah, blah there. Okay. So again, you guys can look at this, different types of symmetry. So you can see some things have symmetry, some things don't. So asymmetrical versus bilateral symmetrical, the different types of tissue layers an animal develops, what type of characteristics it has, whether it has a backbone or not. These are all just ways that we break things down. Here's your common phyla, okay, in order. Here's some example: sponges, jellyfish, hydra and anemone, flatworms, roundworms, segmented worms, different types of mollusks. Here it goes into some of the big groups of mollusks. Here, arthropods and some of the big groups of arthropods, echinoderms, and then, of course, the chordates. What makes chordata chordates? Well, you've got first your invertebrate chordates, and then that would be like the lanceticates and tunicates. And then you have all the rest, which you should probably know these, but if you don't, breaking down the three fish, there's jawless fish, there's bony fish, and there's cartilage fish. So agnantha is its own division of fish. Chondriichthyes, so these would be like lampreys, like the ones that are in the Great Lake as an invasive species, wrecking everything. Chondriichthyes, that would be sharks, rays, and skates. Osteichthyes, that would be all your bony fish that live in the ocean and in like fresh water, the ones that you can go fishing for, and others. Then you get amphibians. Amphibians have to return to water to lay their eggs. So then you have to develop an amnion and an egg that can live not in water. Okay. So then we get reptiles that have dry eggs. We get more advanced hearts. So from a 2-chambered heart to a 3-chambered heart to all kinds of crazy advances. And animals are cool and interesting, and there's lots to learn about them. There's 3 different groups of mammals, which we already talked about. Monotremes like platypus and echidna, they lay eggs, they have poison glands, and they're crazy. And then you have marsupials like kangaroos and possums. And then you have placental mammals. And then the last question is about viruses. Okay, viruses and prions. Prions are crazy, so like BSE. So maybe you guys have heard of mad cow disease. So bovine, spongiform encephalopathy. Okay, it's actually a protein that's produced that gets into your brain and it starts replicating itself. So it's not genetic material. It's literally a chronic wasting disease. It's where proteins in the brain turn against the brain and literally convert the brain matter into protein matter. And then the brain doesn't act like a brain anymore and the animal goes crazy, which is where mad cow disease becomes very scary. Right now, viruses are a huge topic. That's why we're doing this. So, we got coronavirus and the life cycle and why it infects people and how it went through zoonosis from things not humans to humans and all kinds of stuff that we still haven't figured out about this current virus epidemic. All right. I am sorry. I covered a lot of stuff. I wanted to get as much. I have kind of a guilt about the end of the year here. There's so many things that I like to talk about that you guys would have liked a lot more than genetics, because I know nobody loves the genetics chapter. Let's see. Do I need to cover any? I don't think so. Questions. let me stop the screen share. You guys might be seeing there. Okay. Questions from the forum, questions from anything. You guys are back to looking at my mug. Hopefully I covered everything that I could cover in one of our last days of classes. You guys can submit the forms. Thought we'd use the brainstorm as a new way of talking about stuff to open up. Any questions over anything? Look through that notes a little bit better. We maybe one more time, but mostly clean up, fix up, finish up anything that you haven't done yet. If we have another meeting to talk about, I don't know, animals or body systems or something like that, I have so much stuff that we could do, but not enough time to do it. So if anybody has any questions, you can stick around and ask me. If not, yeah, we did it. Anybody have anything? All right. Well, thanks again for coming. Have a great day. Peace out.